So welcome back. Now let's study chapter 15, the diagnosing infections. So in this chapter, we are going to learn three categories of microbial identification techniques. So we have three techniques. So we can use these three techniques to identify the microorganism. So first is the phenotypic technique. So phenotypic technique, so we use this technique to observe the microorganism through a microscope or microscope to observe the morphology of the bacteria, physiology, and biochemistry. The morphology of the bacteria may be the size, the color, the shape of the bacteria. Physiology could be the enzyme, the reactions, um, and the compounds of the cell wall. Okay, so we can detect those for the physiology. Biochemistry. So if the for example, if the microorganism has the capacity of the fermentation, so they can produce acid. Okay, so they can also release carbon dioxide. So we can detect those and product to identify the microorganism. The second is the immunological technique. Uh, immunology means the antigen and antibody reaction. So this technique is used for the serum antibody or antigen detection. So this is called a serological analysis. The third is the genotypic technique. Genotypic means we work on the DNA or RNA because those are the genetic those are the genetic materials, right? So we have to work on the genetic materials. So we can use the PCR or certain hybridization, such as fish technology, to detect the DNA or RNA. So we will talk about those later on. So if we get the data from these three techniques, so we can use this data to identify the microorganism. So as we can see this diagram, so the phenotypic technique is to study on the shape, color, size, or the um, of the compounds of the cell wall of the organelles and the products of the bacteria. Immunology is for the antigen antibody reaction. So genotypic is for the DNA, so we can use PCR. So this is electrophoresis image of the PCR. So genetic means the identification that I use the source and for identify bacteria. So the test can since the development of the technology, so now many devices can be made very small. Also, we can make the process very simple. So many tests can be done by the side. So also many tests can be performed very quickly. So that very reduce the cost and time consuming for those tests. So that provide more tools to detect microorganism to treat the patients. Besides these techniques, we also have the classic imaging technology. These are the CAT scan, MRI, and PETs. So we can use these imaging techniques to detect the local infections in the body. So the phenotypic technique, so we use this technique to observe the trait or the features of the microorganism. For example, we can detect certain enzymes, certain proteins. We also can look at the size or shape. Okay. So the chemical compounds. Immunology actually immunology technology is involved in the antigen antibody reaction. We can use the known antibody to detect the antigen presence in the microorganism to find if this microorganism is a pathogen. So now we have many test kit provided by the companies, such as the ELISA kit or the chip kit or the protein array kit. So we just drop a drop of patient's blood to that kit, so we can detect hundreds, thousands bacteria. So it's very easy. Genotypic method. Genotypic method has its advantage because we don't have to look at the phenotypic feature of the bacterial microorganisms. We just pick a small amount of the samples, so we can run the PCR. So the most important advantage is that we don't have to culture the microorganism. So we just pick the samples such as the sputum, urine, so we can run the PCR. Because some microorganisms are very difficult to be cultured, so this is the biggest advantages for the genotypic method. Now let's do this question. A gram stain of a sputum sample from a patient is an example of what method of microbial identification? So gram staining actually let us to look at the color of the microorganism, right? So the correct answer is C, because the color is one type of the feature, is one type of phenotypic feature. Now let's study section 2. So before we before we identify the microorganism with the techniques, so first we have to collect the specimen, right? To collect the microorganism specimen, so the most important thing is the aseptic technique. So we have to use the steroid tools to collect the specimen. If not, so we will get the specimen contaminated with the other microorganism. So these are the rules to collect specimens. Okay. So we have to collect the samples contains residue microbiota. Okay. Okay. Only the infected site should be sampled. Okay. Note the surrounding area, because there may be tons of normal microorganisms exist on the surrounding areas, right? So we don't want to get those microorganisms to contaminate our samples. As we can see this diagram, so these are the specimens that we will collect in the clinic. Okay. For example, if you suspect the patient has the urinary tract infection, right? So you should take the urine, right? You should take the urine, and not the blood, right? You should test the bacteria in the urine, not the blood or the saliva, right? So we can use the clean catch to cut the urine, or use the catheter to get the urine. So this is catheter to get urine. For the blood, we have to use the needles. Okay, the needles have to be sterilized. If not, so before we take the blood, we have to use alcohol to clean the skin. Okay. For the sputum, we should collect the sputum. Since the sputum is produced by the lungs, bronchi, right? So we should collect the sputum very carefully to avoid the contamination by the saliva, because the saliva contains all kinds of normal microorganisms. So the sputum samples. So the sputum is produced by the lung or bronchi. So the best way to get the sputum is use the catheter, or use the endoscopy to get the sputum directly from the bronchi or lungs. If not, so we can get the sputum after the patient have the coughing. So the sputum can be easily contaminated with saliva, so it can have, so it can be contaminated by the normal bacteria that live in our mouth. So for certain patient, so we need to get the saliva samples so we can use the container and swipe. Also use the swipe to scribe the mouth to get the saliva samples. Urine samples. So we have to use the clean catch to get the urine. Since there are tons of microorganisms lived on the urethra area near the urethra, so we have to be careful to collect the samples. So we have to let the patient pee, okay, and we can give the patient 3 clean catches. So let the patient feel the first catches. So the first catches actually is a dirty catch, because the first word the urine contains contaminated microorganism. So we cannot collect urine from the first catch. So we let the patient fill the second catch. Second catch should be the middle stream. So middle stream urine. So this is the urine. So this is the urine that we want to collect because the microorganism, contaminated microorganism will be washed out by the first rush of urine. So we can collect the second catch. The mucus also, if the urine contains the mucus, so those can be sampled with a swab and sent to a testing at Vir. Skin samples, so we can use a swab, so scrape the skin to expose the deeper layers, because on the surface of skin, so there are tons of microorganisms. So we have to eliminate those microorganisms to limit the contamination. For the wounds, we can swab the wounds or to eliminate the wounds exudate, and then to pick the samples from deeper layer or by punch biopsy too. For the blood, cerebrospinal fluid, or the tissue fluid, so for those samples, we have to use needles. So the needles have to be sterilized, otherwise we will get contamination. Sometimes we also collect the specimen from eyes, ear canal, synovial fluid, or nasal cavities. We can use the swab to get the specimen. If needed, we can take the specimen with the biopsy. Once we get the specimen, we have to put the specimen in the containers or the small catches and label the tubes or catches and seal the catches or the tubes. We can keep those specimens in 4-degree and immediately deliver to the lab for the testing. So here is the sheet for the testing in the medical lab. We can see in this sheet it indicates the patient's name or patient's symptoms. Okay, patient symptoms. It also indicates a water specimen get from the patients. Also, the water test required for this specimen. Okay, and this part may show the results test. So the results tested on this specimen. Now let's do this question. Care should be taken to avoid contamination with what when taking sample from a patient. So I give you 5 seconds. 1, 2, 3, 4, 5. So the correct answer is D. Normal bacteria, normal microbiota, because we want to get the pathogen, right? So the normal microbiota, actually they are not pathogens, okay? So we have to avoid the contamination of normal microbiota. Now let's study the section 3, okay? The section 3 show us the phenotypic method. So so this is so the most important phenotypic method is differential stain. Okay, we can use the differential stain to identify and classify the microorganism. So the differential stain that we have studied in the last chapter is the ground stain and acid-fast stain, right? So this is ground stain. This is ground stain. This is acid-fast stain. We can also use selective and differential growth media. Selective media can only allow certain microorganisms to grow. So we can use this media to kill the contaminated microorganism. So we can use this media for the non-sterilized specimen or the specimen with the contamination. Differentiation media can allow the multiple microorganisms to exhibit different features such as colors, colony size. Also, the may have the gas bubbles, so we can use that to identify the microorganisms. So we use the dichotomous key to identify the microorganism with selective or differentiation differential media. For example, we use the ground staining. Okay, ground staining. When we use ground staining to stain the bacteria, we can get either gram positive or gram negative. Once the bacteria is stained with blue, so the bacteria is gram positive. So we can so next we can do the catalyst test, because the gram positive bacteria can be either catalyst positive or catalyst negative. When we so if the bacteria is catalyst positive, we can look at the cells. We can look at so we can visualize the bacteria through the microscope to look at the shape of the bacteria. If the bacteria is pair chain arrangement with catalyst negative, so that would be Streptococcus. If the bacteria are irregular cluster and catalyst positive, so we can do either aerobic test or anaerobic test. So we can use the reducing media for this test, right? So if the bacteria are aerobic, so that would be Micrococcus. if the bacteria are anaerobic, so that could be Staphylococcus, or Planococcus. If the coccus, if the cocci bacteria are gram-negative, so we can test the bacteria with oxidase and catalase. If the bacteria are oxidase positive and catalase positive, so that would be Neisseria or Moraxella. If the bacteria are oxidase negative, catalase negative, that will be Veillonella. We can also use the biochemical test to identify the bacteria. So since the microorganisms may have different enzymes, so they have different capacity to induce biochemical reactions. So we can detect the expression of this enzyme. Also, for example, if the bacteria has the capacity of fermentation, so we can add the pH indicator in the medium. So once this bacteria fermented the sugar to the acid, so it will change the medium color. So that could be the biochemical test. As we can see this figure. So we can add a substrate such as the pH indicator. So if the microbe has the microbe has fermentase, so the fermentase will induce the fermentation, so it will change the color. If the color has changed, so that will be the positive. If color no change, that means this microbe doesn't have the fermentase, so there is no fermentation. So now many companies provide the rapid biochemical test kit. So that will be a small kit with 20 or 12 valves that contains different substrates. So the nurse or the nurse just need to add the patient's samples to that valves. If the bacteria react with those substrates, so it will cause the color change. Okay, so each valve is labeled by numbers. So if the color changed, so that means this is positive. Okay, so we can see here positive, and positive. Okay, so that means we got the number here for 41444000. So finally we have this code, this numeric code. We can use this code to match with the code database provided by the companies. So on the code database, we can see this is Advancella tada. So we can find this bacteria. So this is how rapid biochemical test is done. Now phenotypic test Now let me introduce the microbacteria growth indicator tube. Okay, so this is one type of phenotypic test. This tube is used to detect Microbacterium tuberculosis. However, the tuberculosis bacteria grows very slowly. So if we want to culture this bacteria for the testing, so we have to wait several days, maybe 10 days, 20 days, So we cannot let the patient wait 20 days in hospital for the treatment. Okay, so we can use this tube. So we add patients samples in this tube. Okay, so the medium contains the fluorescent indicator. Okay, so this indicator is very sensitive to the oxygen level. Since the tuberculosis bacteria is aerobic bacteria, so it consumes a lot of oxygen. It will dramatically induce oxygen level decrease in the tube. Okay, so once the oxygen level decreased, it will let the fluorescent to release a fluorescent light. Okay, so we can use the detector machine to detect the signal change of that tube. So with that tube, we can diagnose the tuberculosis. We can also use the phenotypic test for the antimicrobial susceptibility testing. So we can use this test for the drug test, because once we use the medication to treat bacteria, so many bacteria generate the medication resistance. So we can use this test to detect the medication antibiotics resistance of the bacteria. So if we found the bacteria has high resistance to certain antibiotics, so that will give us the clue to choose which antibiotics to treat that microorganism, right? So now it's widely used to identify the species such as Streptococcus, Pseudomonas, and Clostridium. Okay. Miscellaneous test. For example, phage typing. So the bacterial phage is one type of virus. So this virus is specific attacks the bacteria. Okay, so this attack is very specific because one type of phage can only attack one type bacteria. So we can use this test to identify the bacteria. For example, as we can see in this diagram, so in this plate, so we can divide this plate with different region. Okay, so each region so this plate is growed with the bacteria. Okay, it's growed with the bacteria. So if we put a certain different phages, okay, so so this phage we know, so it can cause the death of the bacteria. So we can know uh, so we can know what bacteria it is. Okay. Now let's talk about the significance of the culture. Okay. So if for example, if for the urinary infections, urinary tract infections, so if the urinary samples contains a few colony of E. coli, so that could be normal. Okay, that could be contamination of the E. coli. Okay, but if the E. coli colonies numbers are enormous, okay, maybe we have hundreds, thousands colonies of E. coli, so that could not be the contamination. That should be infection. That could be the urinary tract infection. So this is one case. Another case is for the tuberculosis. Uh, because the tuberculosis is not a normal microbia that exists in our body. Okay, so if we collect the sputum samples, even we only find one single colony of Mycobacterium tuberculosis, so that's it. So this is tuberculosis, because normally the people don't have tuberculosis in their respiratory tract. Okay, so this is the second case. Also, when we take the cultures such as the blood cultures, urinary cultures, so it's a good idea to repeat to do the cultures repeatedly. Okay, um, so that will confirm our identification. Another things we have to know about the culture. Okay, um, the culture actually is relative time consuming. Okay, at least the culture will need 18 to 24 hours. For certain bacteria such as tuberculosis, so it may require a much longer time than that. So so that's why in the clinic, however, we cannot let the patient wait for 20 hours without treatment, right? So in the clinic, so the doctor will give, so the doctor will collect the samples for the cultures, for the bacterial cultures. In the same time, they will treat the patient with broad, so with broad spectrum antibiotics. As we can see in this diagram. Okay, so the doctor can assume a diagnosis based on the symptoms of the patients, and then give the patients with treatment such as antibiotics. If so if it works, so the um, so the so so the patient get better. So that means the antibiotics that used is correct. Okay. However, if the patient doesn't get better, so maybe we can get the culture. Okay, bacteria culture. So we can, since we don't know which bacteria cause the problem, so we can do several cultures, such as the selective differential growth, biochemical testing, susceptible testing, and miscellaneous testing. Okay. Now let's do this question. You have obtained a fecal sample from a patient. What type of media should you use to grow a suspected pathogen from this sample? So I give you 5 seconds. 1, 2, 3, 4, 5. So the correct answer is E. Okay. All of these choices are correct because the fecal samples contain a lot of microorganisms. So it's difficult to use only one method to isolate the pathogen. So it's a good idea to use all of this technique method to detect the pathogens. Now let's work on the section 4. Section 4 talks about the immunology method. This method is also known as serology technique. So this is the in vitro testing of the serum, urine, or body fluid. Okay. Since the immunology is the antigen-antibody reaction, so this test can give us the idea if the patient have been exposed to the diseases before or not. So the antigen-antibody reaction may not be visualized by the naked eye. Okay. However, we can we can visualize this through the microscope. Okay, and identify the reaction. As we can see in this diagram. So the patients we can so if we don't know the if we get the patient's serum, we don't know if the patient has been exposed to a certain disease. So we can mix the patient's serum with known microbial antigen. So if the agglutination happened, so that means there is antigen-antibody reaction. So that means the patient has been exposed to those disease. We can use this method to detect to identify the microbe as beer. We can use antibody of we can use the antibody of a known microorganism to detect the unknown colony of the microorganism. If there is agglutination, if there is aggregation happened, so that means this unknown microorganism is so that means the unknown microorganism is what we studied. Okay. So we can see this diagram. So the immunologic method can induce agglutination because both antibody and antigen will be soluble in the serum or the liquid. Okay. However, once they linked together, combined together, so they will be insoluble, so it can cause agglutination or even cause precipitation. So once we found the agglutination of the precipitation, so that's positive for the immunological test. Because immunological test also contains immunochromatography and Western blot, fluorescence antibodies test, ELISA and INVIVO test. INVIVO test would be the TB skin test. Also, the flu shot. Flu shot could be one type of INVIVO test at beer. So we talk about this later on. So the immunology test actually is antibody and antigen reaction. So the binding reaction so the binding reaction could not be visible. Okay. Actually, so the antigen and antibody are soluble in the solution. So the binding can cause the clumps agglutination. So that will be seen through either by the naked eyes or the microscope. So the agglutination reaction can happen either one antigen is linked by several antibodies to form insoluble aggregates. Okay. So the agglutination. So if the antigen are very huge, such as if the antigen are so if we detect the antigen in the cells, okay, so the cells could be so the cells size is very huge. Okay, so if the antibodies link the antigen on the whole cells, so they can link the whole cells together, so that will cause agglutination. So the agglutination will forms visible clumps of the cells. Okay, so we use this mechanism to do the blood typing. Okay, to do the blood typing. Also, we can also use agglutination for the via Felix reaction to detect Ricus infections. Precipitation. So if the antigen is a soluble molecule, so the precipitation is more difficult to visualize because it can be disturbed by the liquid media. So probably we can use the microscope to visualize carefully. Here is one type of agglutination test. So this is 96 beer plates. This is also can be used as microarray or microchip test. So in each small tiny wheels, we can link with certain known antigen or antibody. Okay. So and then we can add the patient's samples. If the patient samples contains the antigen, that antigen react with antibodies, so we can know, so we can identify the antigens. So each will contain different antibodies, so we can detect different microorganisms. Immunochromatography. So the one example of this test is a pregnancy test. The pregnancy test actually is a plastic cartridge. Okay, it contains porous material to allow the diffuse of the urine samples. On the materials, actually, an antibodies for the HCG is coded to that material. Because we know that once the female is pregnant, so the urine HCG level is very high, right? So once the urine diffused on the cartridge, so once the HCG reach to the antibodies, so the HCG will be so the HCG will bind with the antibodies and then induce the color change, So as we can see, this is control. This is test. Once we find the band in this part, so that means the female is pregnant. So we can load the urine samples in this sphere, and then urine will diffuse to this direction and react with the antibodies. Antibody titer. So we can use this technology to test the efficiency of the antibodies. So we can dilute the antibodies and then to react with certain antigen. The more so if we dilute the samples, so if we make the more dilution of the samples, however, the samples can still react with antigens. So that means the titer of the antibody is high. Okay, that means the efficiency of the antibody is very high. So this technology is also used to detect the autoimmune disorders and past exposure to the certain diseases. Serotyping. Serotyping. So this is one part of immunology technique. So this is also antigen-antibody reaction technique to identify certain bacteria, certain bacteria and antibodies in the serum. So we can use this technology to detect the specific bacteria. For example, if the bacteria contains the capsule, flagella, or the specific cell wall, so we can use this technology to detect the antigen on this specific bacteria structure. So this technology can help us to identify the bacteria such as Salmonella because Salmonella has flagella. Okay. and for the pneumococcal and streptococcal, because those bacteria contains the capsule, okay, contains the capsule, so we can use this technology to detect certain species. Western blot. Western blot is widely used by the research and hospital. Okay, when we do the Western blot, so first we have to lysate the samples, or lysate the cells, or lysate the bacteria. Okay, so we have to let the protein in the cells to be released, and then we can load the samples to a gel. Okay, so this gel is called SDS gel. SDS gel. And then we run the electrophoresis to separate the proteins on the gel. Once the proteins is separated on the gel, so we can transfer the proteins in the gel to a membrane. Okay, to a membrane, and then so we incubated the membrane. So this membrane filter. Okay, so this is the filter. Actually, is in so this is one type of paper. Okay. So we can incubate the membrane with antibodies. Okay, antibodies. So actually, we incubated with two antibodies. The first antibodies is called primary antibodies. The primary antibodies is specific to detect the protein that we want, and then we incubated with the secondary antibody. Okay, secondary antibody. Secondary antibody is the antibody labeled with radioactive or the fluorescent. Okay, so the second antibody can give us the signal to detect the bands. So once the second antibody is conjugated to the primary antibody, so we can read this membrane, this filter membrane with specific devices, so it can detect the is radioactive or the fluorescence released from a secondary antibody, so we can get the signal. That we can, so if the bands is visualized, so that means so we detect detect what we detect the proteins that we want. So we can see the protein is transferred to the membrane. Okay, this is called blot. So this is the primary antibody. Is specific to link with the protein. So this is the secondary antibody. So secondary antibody is conjugated with is radioactive or the fluorescent to give us the signal. Now let's talk about the immunofluorescence testing. Since this is the fluorescence testing, that means the antibody is conjugated with fluorescence, so it can give us a fluorescence signal. So we have direct testing. Direct testing, so this means we want to test a certain antigen. So antigen is fixed to a size or a membrane, and then so we incubate the size or membrane with the fluorescent antibodies. If the antibody react with antigen, so we can get the signal. So we use this test to detect the syphilis, gonorrhea, and meningitis. As we can see in this diagram, so we use the fluorescent antibody detect the syphilis. So the syphilis shows the green colors. So the green is the fluorescent signal. Indirect testing. Indirect testing, so we can use known antigen to detect so we can add a known antigen to the serum to detect if the antibody exists in the serum. So if then we can add fluorescent antibody. So the if the if the patient's serum contains antibody to the known antigen, and then so the antibody will conjugated to the known antigen, and then we can add this fluorescent antibody to conjugate it with the antibody that conjugated with the antigen, so we can see the signal. So this technology is also used to detect syphilis and several viral infections. So the indirect test is to detect antibody. Enzyme-linked immunosorbent assay is called ELISA. It's widely used in clinic and research. This is also called as sandwich testing because we use plastic plate. So we coat the plastic plate with the antibodies, and then so so we have indirect ELISAs. Okay. So we coat the plastic plates with the known antibodies, and then we add the patient's serum. If the patient's serum contains the indirect, so we use the known antigen to coat the plate, and then we add the patient's serum. If the patient's serum contains the antibodies to certain diseases, so these antibodies will be conjugated with the coded antigen. Okay, and then we can add secondary antibodies to give us signals. Direct ELISA. Direct ELISA, that means we use known antibodies to coat the plastic plates, and then we add the patient's samples to the plates. So if the patient's samples contains the antigen that we study, so the antigen will link to the antibodies that code on the plastic plates, and then so we can wash out the patient's samples, so we can add a secondary antibody to give us to give us the signals to show this reaction exists. So we can use this ELISA to detect the all kinds of virus infections and toxoplasma infections. So and then we can put this plate in specific detecting devices to read the signals released from the secondary antibodies, so we can get results. As we can see in this diagram, okay, so this is indirect ELISAs. So we use the known antigen to coat the plastic ware, and then we add the patient's samples. So if patient samples contains the antibodies for certain diseases, so the antibodies will be conjugated to the antigen. Then we wash out the patient samples, and then add the secondary antibodies. And then secondary bodies will change the colors or release the light signals so we can detect it. So this is direct ELISA. We use the known antibody to coat the plastic ware, and then we add patient samples. If patient samples contains the antigen of certain microorganisms, so the antigen will be conjugated to the antibodies that code on the plastic, and then we add the secondary antibody. The secondary antibody will change the color or either change the color or give us that signal so we can detect this reaction. In vivo test. In vivo test, so the good example is the tuberculin reaction. Tuberculin reaction actually is used for detect if patient has been exposed to tuberculosis before. The people isolate purified protein derivative from the tuberculosis. So this is one fragment, one protein fragment of tuberculosis. So this tuberculosis have all the informations of the antigens of the tuberculosis. Then the nurse can inject this fragment, this protein fragment to the patient's skin. Okay, so if the patient this fragment, this protein fragment is recognized by the patient's T cells. Okay, because tuberculosis is the immunological diseases. Okay, the T cells will be involved in killing tuberculosis. So if T cells can recognize this protein fragment, so the T cells will migrate to the injection site and attack this protein and cause the redness and raise the lesion. After 48 or 72 hours. So after injection, after 70 hours injections. So if we found these lesions, so that means this patient has been exposed to tuberculosis. So once we studied the immune test, so we have to know the features for the immune test. One feature is the specificity. Since the immune test is antigen antibody reaction, so that means so this is specificity. Okay, because one antibody is one antibody is only reacted with one antigen. Okay, so this is specificity. For example, we use the PPT for the tuberculosis test. We cannot use this for other microorganism test. Okay. Another feature is sensitivity. Sensitivity means efficiency of sensitivity means the conjugation efficiency of the antibody to the antigen. Okay. So the problem of sensitivity is that this test can give the false positive. Okay, false positive. The sensitivity means even very small very small titer of antibodies can still react with the antigen. So if the quantity of antibody is very low, however, it can still react with antigen. That means the sensitivity of the antibody to that antigen is very high. Now let's work on this question. Direct ELISA detect what, and indirect ELISA detect what from patient samples. So the direct ELISA actually detect the antigen, right? Detect the antigen. So the indirect ELISA is 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 used to detect antibody. Now let's work on section 5. Section 5, so we can learn the genotypic test. The genotypic test, we have several techniques that we use for this test. The first is the PCR. Second is the hybridization and genome sequencing and gel electrophoresis. PCR means the polymerase chain reaction. So this technique is invented in 1980s. It is used to detect DNA and RNA. So now we have several different PCR. Now we have the real-time PCR. This is also called as qPCR. Q means quantitative, because the PCR actually uses some cycle to amplify the DNA or RNA. So if the DNA or RNA concentration is relatively high, so it will get so it can give the signals even in the very early sum cycle. For example, so we run 30 sum cycle for the amplification. So if the DNA concentration is high, so maybe we can get the signals even at 10th sum cycles. So we get the 10th numbers for this DNA. So that means after the reaction, so we can get all different numbers for different DNA RNA. So that is quantitative PCR. Because these numbers, so we can get these numbers during the running. So this is called real-time PCR. Multiplex PCR. So we use the primer for the amplification of the DNA RNA. So we can use the different primers for different organisms. So we can mix those primers together to detect the patient's samples. So if we get certain primers, so if we get reactions for certain primers, that means we know certain microorganisms are involved in the patient's diseases. Actually, so the multiplex PCR is the real-time PCR, because it's difficult to run the multiplexes PCR with the normal PCR. So we have to run the real-time PCR. So it will give our the numbers for different primers. pan-bacterial qPCR. Pan means universal. That means we mix all kinds of primers. So this is as the universe primers. These are the primers of 16 small ribosome RNA. So it will amplify all the bacterium RNA, so we can get many data for the bacterium. So so this is also qPCR. Hybridization is the technique to detect the pathogens. So this technique requires the probe. The probe actually is a small segment of DNA or RNA. So this segment of DNA or RNA is conjugated with fluorescence. So we can incubate this probe with the samples. So if the probe is conjugated with the microorganism, so we can know what what microorganism it is. Okay. Another technique for the hybridization is the fluorescent in situ hybridization. It's called FISH. The advantage for this technique is that we don't have to so we can use this technique to detect the intact cells. Okay, intact cells. So we don't have to prepare the cells. We don't have lysis the cells. So we just fix the cells, and then we can incubate the cells with probe. Okay. So this technique gave us the image of the samples. Okay. So we have to use the microscope to analyze this image. Okay. So this can be used to confirm the diagnosis or to identify the microbial components within a biofilm. It can also be used in the cancer diagnosis to analyze the medication resistance of the cancer to the chemotherapy medication. So this diagram shows the image of fish. Okay, so we use the probe. That's nucleic acid fragment specific for the Staphylococcus aureus. Okay, then we can code the samples on the slides, and then we fix the samples. So we can incubate the samples with the probe. If the probe is conjugated to the Staphylococcus RNA, so it will give us the fluorescent signal, so the green colors, as we can see here. So we can visualize the slides with the microscope. The microarray. Microarray is a very new technique. Since this is array technique, so it is also called as chip. Okay, it's chip because it can detect hundreds or thousands samples in one time. So actually, the array plate is a multiple vias plate. So each vias is coded with specific primers. Specific primers for one specific microorganism. And then we load the patient samples to each via, and run the test. So once the antibody is reacted, once the antibody reacted to the patient samples, so it will give us fluorescent signals. So we can detect these fluorescent signals with a reading machine. As we can see this diagram. So this is the microarray plate. So each pair is coded with specific primers. So the primer, then we load the patient samples if antibody antigen reaction happens, so they will release the fluorescent signal, so we can detect these fluorescent signals with the computer and devices. Whole genome sequencing. So we can use the whole genome sequencing to get all the genome sequence of the microscope of the microorganism. So we can use this technique to study on the drug resistance. So in the beginning, whole genome sequencing is was very expensive. It cost about $10,000 to sequence just one chromosome. But now it's relatively cheap. It costs $1,000 to sequence one chromosome. So we can use this technique to sequence everything, okay? And then we have the pulsed field gel electrophoresis. So this is used for the microbial fingerprint. So we use this technology. Actually, we use this technology to analyze the DNA. Okay, since the DNA is very long, so we can cut the DNA as several relatively small fragments and run the electrophoresis. So this electrophoresis is very special because the devices apply alternating voltage to the gel, so can from three different directions. So it can separate the similar size DNA. Okay, the similar size DNA. So once the DNA is separated, so we can get the so we can detect the gel with the fluorescent. So the fluorescent will show the bands of the DNA. So we can use this band to match the database of the CDC to find the to identify the microorganism, as we can see here. So we get the bacteria samples. Okay, we lysis the samples to release the genome. Okay, release the chromosome. Since the chromosome is very long, the DNA is very long, so we have to use the restriction enzyme to cut the DNA to small pieces. However, the small pieces is still very long, so we can use the pulsed field gel electrophoresis to separate those fragments. And then we can get this image, this DNA bands image. So we can compare this bands image with the database in the CDC to identify the microorganism. Now let's work on this question. First, in situ hybridization requires the pure culture of organism obtained from a patient sample. So I give you 5 seconds. 1, 2, 3, 4, 5. So actually, it is false. So the advantage for fish technology is that we don't have to prepare the samples. Okay, we just fix the samples and incubate the samples with the problem, so we can get the image. So that's it. Now let's work on the section 6. We have talked about the microarray. Okay, so the microarray is called a chip. Once the microarray is invented, so the microarray plate is very big, was very big. Okay, so with the development of technology, so now the microarray plate is made smaller and smaller. However, it can detect more and more samples. It's like this. So advantage is that so it's very easy to use. Okay, so the we just load the patient samples, so it can detect thousands or 10,000 microorganisms. Also, the instrument is very small, so it's very good to be used in the developing countries. Okay. Now let me introduce the mass spectrometer. It's also called a multi-top. Okay, it's the technique can detect the molecule very fast and very accurately. Okay. The mechanism of this technique is to load the samples on the tip of the on the metal tip and use the laser to shoot on the samples to ionize the samples. Okay, to let the samples degraded to iron. Then the devices can detect the so the ions with to detect the ions by their mass to charge ratio. So with this with the data, so they can identify the proteins, lipid, or the proteins, lipid, or sugars. Sugars composition of the microorganism, and then to identify the microorganism. Imaging. The imaging technology includes CAT scan, MRI, and PET. So we can use this imaging technology to identify to find the localized infections in the deep tissues without surgery. Then it can give us guide to take the samples from the localized infections. Then based on the samples, so we can find what microorganisms cause the infections. So with the development of technology, so we have more and more new diagnostic strategy. Okay, so now the scientists can scan the antibodies for more than 1,000 strains of the virus uh, to discover which pathogens cause the problem for the patient. Now let's work on this question. Which of the following technologies detects microbes via their protein fingerprint? I'll give you 5 seconds. 1, 2, 3, 4, 5. Yes, so that's a mass spectrometer. Mass spectrometry. So here is the appendix. So you guys can feel free review the contents of this chapter based on this appendix. So this ends.